In 1952, two scientists, Hershey and Chase, were studying bacteriophages. A bacteriophage is a virus that attacks a bacteria by attaching to the surface and injecting some substance into the host. Their aim was to see what was being injected into the bacteria by the phage. Hershey and Chase approached their experiment by labeling the two parts of the bacteriophage, the protein and the DNA. They used 35S to label the protein, and they labeled the DNA with 32P. The sulfur and the phosphorus are taken up into the protein and the DNA so that they can be identified later on in the experiment. Hershey and Chase prepared two separate batches of the phage and mixed in labeled bacteriophages with E. coli. After allowing the E. coli to be infected, they used a blender and then centrifuged the cultures to remove the protein coats and any remaining phage from the cells. This resulted in the supernatant and the pellet. During centrifugation, lighter materials will remain near the top of the tube and form the supernatant. The supernatant is the liquid portion that includes the phage parts. Heavier materials will move toward the bottom of the tube and form what is called the pellet. The pellet contained everything that was remaining in the cell. In the supernatant, they found that the 35S was remaining, while the 32P was in the pellet, meaning that it remained in the cell. What this showed Hershey and Chase was that the material that the phage injected into the bacteria was in fact the DNA and not the protein. This allowed them to conclude that the DNA is the genetic information in the bacteriophage.